Appreciate the opportunity to present to you guys today. This is my first experience with the North American PRRS Symposium, and there's a reason for that. It's as I'm classically not trained as a virologist, and today I'm not going to talk to you about a virus. I'm going to talk to you about a bacteria. So, knowing that I was last, and right after a lunch, I actually had to be real vague with my title, so I didn't have people running to the door. So today I'm going to talk about what we've done over the last several years, actually to build a specific product. You can see it here. It's called Fostera PCVMH. And when we started this program several years ago, we were looking for a vaccine to deliver to the industry that gave three things, and I'll talk about those in a little bit. But before I get to that, we could have taken the easy route forward and actually just taken some of our existing products, and we have a long list of products in this particular area, put them together, but there were several issues that came up. One I'll talk about specifically. I'll spend probably most of the time discussing it today. The other is that in most of these cases, the formulations weren't compatible. So what we were looking for was a vaccine that had a safety and efficacy profile of the existing products. Our vaccines work very well. They have a good safety profile, so we were looking for something that was equally as good as those, if not better. The hard part we found out was coming up with the convenience. So what we wanted was a single-dose product, so you don't have to administer it to pigs one time, that comes in one single bottle, so you don't have to worry about taking one bottle and mixing it with another bottle at pig side. So what we were looking for was a one-dose, one-bottle format to deliver to the industry for convenience and, of course, not sacrificing any of the safety or efficacy profiles. So we'll foreshadow a little bit. We were successful in doing that, again, and that product is called Fostera PCVMH. So the major technical problem that we ran into very early in the program. So this is a rough flowchart of how we actually make, first of all, mycoplasma high pneumonia antigen. And again, this is a combination vaccine, PCV2 and mycoplasma high pneumonia. One of the key components for the M-Hio antigen is actually porcine serum. And that serum is sourced through commercial vendors. And and those pigs are exposed to natural agents. Some of them, in fact, I think a good deal of them are actually immunized. So you can imagine they have antibodies in that serum, and some of those antibodies are actually specific for PCV2 antigen. And so when we actually mix the M-Hio, and the circles here represent the mycoplasma, and these are depicting the PCV antigen. And so when we put those things together, the antibodies we found bound to the PCV antigen, and actually when we actually go test that in a critical quality control assay to release that vaccine to the market, we were not able to detect the virus in our potency assay, which is, in this case, it was ELISA-based. And so this ended up being a very major technical problem. Early on, we thought perhaps that it was potentially having an effect on efficacy as well. But since that time, we found out that it does not. And also know that there are a couple of competitors out there with a vaccine that's similar, and their efficacy profiles work just fine as well. So this truly it's a in vitro artifact that we had to solve. So, first thing out of the gate, we'll do this easy, right? We'll grow up our bugs. We'll separate them with a very simple liquid-solid separation technique. We'll use a centrifuge, an old-fashioned device. So what we find out is if we do that, and we wash them appropriately, we can find that, and these are representing the relative levels of anti-PCV2 antibodies. We can actually, most of the antibodies, as you would expect, they're soluble, end up in the supernatant fraction, and very little ends up in the cells. The problem with that was, at least initially, is that if you look at the M-Hio antigen, a lot of the antigen, and we have data that we looked at 3 different indicator antigens. A lot of that antigen ends up going with the antibodies in the supernatant. But we wanted to make sure that this vaccine was compatible, so we actually ended up throwing a lot of those away and just keeping the pellet of cells. And so I've got this depiction here of showing that 90% of the antigen is actually in the supernatant. If you take this little tiny cell fraction, you can actually get enough antigen, and if you put enough of that in the vaccine, you can actually theoretically get it to work. Theoretically. So what we did then is we had theoretically solve this problem. We made a vaccine. We went out, we ran a study, and this was something right in the middle of the development program, and you notice that we called it a spectacular failure. This is a study that was really going to take us deep into development. So this is a study where we actually taken liquid-solid separation or centrifuge cells, no supernatant, but centrifuge cells, and we actually put it at 3 different levels in a study at what we call low, medium, and high levels. And what we found is that in each case, we did not see a significant effect on the ability to protect these animals from a virulent challenge. And so we stepped back and went, what happened? And we found out later what happened, but before we get there, we were trying to figure out an approach that we could actually come up with an alternative to centrifugation to remove these antibodies and still keep the antigens in the vaccine. And so a bunch of really smart people got together in a room. We came up with a bunch of different potential ways to do this. And what we found is that for the most part, with the exception of one, which is I'll spend the rest of the time talking about, none of these were places that we wanted to go. So we could, first of all, try removing the antibodies from the swine serum and grow the mycoplasma, and then take that antigen and combine it with the PCV. No antibodies, we should have good compatibility. The problem with that is that when we started the program, we were challenged to stay with the M-Hile production model that we currently use. We had, since 1992, really good results in the field and a really solid vaccine called Restasure. And so we were challenged to stay away from that. We also found out, we didn't really listen very well as scientists, we tried going ahead and treating some of that serum, and what we found is that treating that serum had a negative effect on the mycoplasma production model, and actually had a negative effect on growth. I don't think I'm going to spend much time talking about growing M-Hile without serum. I think there have been people that have spent almost careers trying to figure out how to grow mycoplasma without serum, and it's just not possible. We also thought about trying an alternative source of serum to grow the M-Hile, instead of using swine serum, using something like horse serum. But again, we felt that we didn't pursue that very much because we didn't want, heterologous serum going into the animals. We didn't want to affect the safety profile of our vaccine. We didn't know what effect it would have on efficacy. And again, we didn't want to affect the efficacy profile of the vaccine as well. And again, we were challenged to stay with that Restasure-like M-Hile model. There were some other things, I'm going to jump over this one for obvious reasons, but there were some other things we tried. We actually considered trying to source CDC swine serum. You can imagine that would be very expensive to do, so the cost of our vaccine would go way up to the point where it wouldn't be economical for producers to buy it. We actually looked at a single vial format that actually had 2 compartments in it that all you had to do was push them together and the 2 liquids would mix together. That's actually something that's used in certain human medicines, but if you imagine trying to have hundreds of thousands of doses of these things in a producer, it wouldn't be very convenient for the producer to actually use that vial format, so that quickly got pushed aside. We considered for a while going to the 2-bottle format, but again, we were challenged by our commercial organization and really the industry to stay with that single bottle format. We also had a team of folks actually working on an alternative potency assay that wouldn't be victim to the effects of having the anti-PCV2 antibodies in there. But lo and behold, we actually had a proposal by one of my scientists to use protein A. I'll talk a little bit about protein A here shortly. But one thing that this particular solution to the problem required was we actually needed to remove the cells, and that's certainly new to the M-Hile vaccine arena because all of the vaccines out there right now are all bacterians, and so we weren't exactly sure whether this was even going to work. But lo and behold, it does. And before we ran the study I'm about to show you, we actually did some in vitro assessments, and we think we know why it works. And so what we did, these are 3 monoclonal antibodies to P65, P46, and P97. And we took some of our fluids. And what these are molecular weight standards. This is the whole fluid, so this is the bacteria plus the supernatant. This is a cell-free supernatant that we just pelleted the organisms out, collect the supernatant. And then the last column here is actually the cell pellet resulting from that. And two things about this is we actually washed these cells extensively because we found that we had to do that in order to get rid of sufficient levels of antibody. I'll also point out that this is actually a 10x concentration of those cells. So this is how this is one of the things we use to determine that only 10% of that antigen was actually going with the cell pellet. So this, you can imagine, is actually a 10% less amount of antigen by volume going into this particular well compared to these things. So what we concluded from this is that as the mycoplasma are growing, they're reproducing, they're actually spitting their antigens into the supernatant. And so, and apparently those antigens that are in the supernatant are the ones in our bacterin vaccines, which contain both cells and supernatant, that are giving us our efficacious profile. So with this information, there's our centrifuge tube again. With this information, we actually move forward to design a study, which I'll talk about shortly. But before I get to that, I thought I'd talk a little bit about protein A, and this is the unique, the modern technology part of this particular vaccine. Protein A is a virus factor of Staph aureus, a well-known nosocomial infection. What it does is it interferes with optimization and subsequent phagocytosis by binding antibodies, which is important. Its activity was first described in the 1940s, and in the early to mid-60s, it was actually, the individual protein was described. I think it's about a 42 kilodalton protein. And it was actually found in fraction A of an extract that they did in that work back in 1964. In the 70s, they were actually able to purify this large scale. If you guys have ever purified antibodies, you probably used protein A, but you probably used it in a little purification format as opposed to at industrial scale. And these excretor proteins, normally it's expressed on the surface of Staph aureus, but some of the excretor strains actually had mutations where they were spitting it out into the supernatant, and it could be purified from that. The gene was cloned in the mid-80s, and in the 90s, actually, that recombinant protein A was coupled to Cephalus, and that became available. And that is actually used to produce a lot of human biopharmaceutical agents, and I'll talk about those here in a little bit. I put these two figures in here just to give you an idea that this is, and quite frankly, this whole slide, give you an idea that this is a molecule that has been well studied, especially in the human medicine arena. We've gone from describing to actually a biotechnology use for this particular material. This is a crystal structure, and this is a ribbon structure showing one of the molecules in its natural state and one in its lowest energy form. But what this stuff is typically used for, and I'll skip ahead one slide, is actually to purify monoclonal antibodies that go into human medicines. And you may recognize some of these, and I just pulled this actually off of Wikipedia. Notice that it stops with down at the eye, and so there's a whole bunch more of these that are available. You may recognize one of these, Humira. That's a TNF, anti-TNF monoclonal antibody. So this material it's well described. It's well used in the industry and for human medicines, and there are dozens, if not, well, at least dozens of products that are out there that use this particular material. The reason it hasn't caught on in the animal health arena yet is because until recently we don't have any of those. And number two is this stuff is really expensive, so it'd be difficult to actually produce a vaccine, especially for livestock that's still cost efficient. I'm glad to say we were able to figure that out. So this is what it looks like. It's actually this is actually the Cephalus, and the molecule has been engineered to have a cysteine on its terminal end. And through a thiol ester bond, ether bond, it's hooked to agarose. And what happens is you actually pour a conventional column, and as you percolate your material through there, it will actually bind antibodies. I was able to find a paper that described that they think that this particular molecule binds 2 antibodies per protein A molecule. The molecule is actually, the way the recombinant commercially available material is made is it's actually directionally linked, so it actually maximizes binding, and it has very, very high binding affinity. It has 5 active domains, and each one of these have the capability of binding an antibody. It primarily binds in the constant region of the FC portion, although one of the domains, I can't remember which one, but it actually will bind at the hinge region as well. So again, point here is this stuff has been around a while, works really very well, and importantly, for the success of this particular vaccine, it binds porcine serum fairly well. And again, this is just relative binding. And it binds it with very high specificity and very high affinity. With that in mind, and keep in mind, we were working on these processes in the lab at about the same time, or right after that we had our spectacular failure. And so we were trying to figure out what was going on with our particular antigen fractions. And so we set up this fairly elegant study in which we looked at a bunch of different combinations of centrifugation, concentration of the antigen. We actually looked at centrifugation and heat because we had observed that if you heat the mycoplasma, you can actually release more of the antigen. Note that there is one treatment in there where we actually collected the supernatant and treated it with the protein A material. And then we had our positive and negative controls. These are colored for a reason. Anything that is purple is compatible with PCV2. Anything that is black is not compatible. And that becomes important for this next slide. We put together a Venn diagram. So again, anything in this circle is PCV compatible. Anything over here we observed that was efficacious based on this study. And what we found out is that one of those treatments actually gave us both a compatible as well as an efficacious antigen, and that was the cell-free supernatant treated with the protein A. So I'm not going to go any more data. I could show you dozens of pig studies. In fact, here are all the studies we've done. These are the studies we used to register the product. I'll note that we have both a single dose as well as a 2-dose product. You can either give it once at 3 weeks of age, or you can give it one 2 ml dose, or you can give one 1 ml dose at 3 weeks of age and a follow-up 1 ml dose at 5 weeks of age. And again, these are registration studies. We've also run the vaccine is effective in the face of maternally derived antibodies. So, what have we done? We've actually come up with a new paradigm for an immunoantigen 1. There are no mycoplasma cells in there. We actually remove them. And the reason we remove them is that in order for us to percolate this stuff through that protein A, you have to remove those cells, otherwise that stuff will follow. And you don't want to follow a $500,000 piece of essentially a filter. So it's a non-bacterian approach. We believe that by removing the cells, we can actually get, it contributes to the overall good safety profile of this vaccine. I know in our field safety studies, we had very, very low incidence of injection site reactions, well below 1%. So it is a very smooth vaccine. I will note that the reason I'm able to talk to you about this today is it is patented. We have got patents granted and pursuing more. And it is compatible with PCV2, and it is a protein-reduced vaccine.